I am going to hand you over to Karen. Thank you very much.
Thank you, Alison. Right, so first test is sharing my screen. Is that okay?
That's great. Thank you, Karen.
Well, yeah, thank you very much for the invitation to come and speak tonight. Hopefully you'll see by the end of my presentation why it's really good timing that you've invited me right at this very moment, because it is very timely and we're at a really, really exciting stage of our research. So today I'm going to be talking about some work really that we've been doing at the University of Leicester for about 20 years. And I'm going to be describing how we've taken resveratrol, which is a natural compound found in red grapes, red wine and peanuts, from literally the bench in our lab to a large clinical trial across England and Wales. So that may not be clear from the title of my presentation, but I'm going to be talking in general about cancer prevention and why we need cancer prevention. And then I'm going to focus in on the area that I work on, which is the use of therapies to prevent cancer. Talk about aspirin and how that we know that works and how that has fed into my own research. And then the bulk of the rest of my talk is going to be on resveratrol and our work in Leicester, where I'll describe how we did some clinical trials in our local volunteers and patient population at the Leicester Royal Infirmary, and then all our lab work that's gone alongside that and how it's led to this big clinical trial. So yeah, so it's going to be a bit of a description of 20 years work. So just to start then, I'll give a bit of background on cancer, potential causes of cancer and why we need to do a better job at preventing it. So this is a news article in the BBC website from 2013. And at that time, they were talking about Macmillan Cancer Support, who had predicted that by the year 2020, 1 in 2 of us will develop cancer during our lifetime. So we're now past that time, and about half of the UK population will develop cancer. And the cancer burden is going up. So these are the figures for Europe. In 2015, the number of new cases per year was 3.6 million. And by 2035, that is predicted to go up by 20% to 4.3 million. And deaths also are going up by 30% over that time frame. And this increase is entirely due to the fact that the population is growing and aging. And on top of this, there's likely to be extra increases due to exposure to risk factors and due to behaviours and certain lifestyle choices that people adopt that might put them at higher risk. So that these numbers are likely to go up, not down. And at the moment, the annual UK cancer costs in 2020 were 24.7 billion. So it's only now becoming apparent to governments and organisations across the world that no single country can afford to treat their way out of the cancer problem. We absolutely have to be putting a lot more emphasis on prevention, because it's just not viable to keep going like this, because the more people getting it and the cost of treatment is just going up and up. So we absolutely have to do more for prevention, not just because of the economic cost to a country, but obviously the effects on society, the effects on families, and the effect on the patients. It goes without saying that it's much better to try and prevent cancer than to wait and cure it. So this is an article from 2015 where they looked at factors in the UK that potentially might be associated with increasing our risk of cancer. And they identified 14 potentially avoidable risk factors. And they're divided up, as you see on this graph here, with tobacco smoking being the highest cause of cancer. But together, these factors account for about 40% of cancer cases. So what that's saying is that about our lifestyle choices and our behaviours potentially are contributing to 40% of cancers. And I've highlighted the ones in yellow, which are all associated with our diet and physical activity. So you can see that the second highest contributor to cancer risk is being overweight and obese, and then drinking alcohol, not having enough fiber in your diet, eating too much processed meat, and insufficient physical activity. They're all major factors in contributing to our risk of cancer. And then on top of that, you've got things like infection, air pollution, use of oral contraceptives, so they also play a role. And these are all things that we can change. It's not necessarily easy to change them, but they are things that we can modify to reduce our risk. And I just want to spend a bit of time talking about obesity, because that feeds into some of the work that we've done with resveratrol that I'll describe a bit later on. And obesity is now recognised in the UK as the second highest contributor to cancer, second only after smoking. And because 6 in 10 adults in the UK are overweight or obese now, this problem is going to get bigger, not smaller over time. And these cancers listed here are the ones that are linked with obesity. So there's about 13 cancers where we think there is an association, or if you're overweight or obese, you're at an increased risk of developing these cancers. And in addition, I don't think it's very widely known amongst the general public that having type 2 diabetes can also increase your risk of certain cancers. And so the ones in yellow with arrows, such as breast cancer, liver cancer, pancreatic cancer, bowel and uterine cancer, they are also independently linked. If you've got diabetes, you're also at a higher risk of developing these cancers. And that could be because the risk factors for developing diabetes and cancer are similar in terms of obesity and other diet and lifestyle factors. Or it could be that having diabetes contributes to the causes of cancer, and that isn't really known yet. There's still a lot of work needs to be done around that area. But essentially, our metabolic health and whether we are overweight or obese can influence our risk of cancer. So, as I said, as a whole, about 40% of cancers are thought to be preventable through changing our lifestyle and behaviours. But this isn't the same across all cancers. So the cancers can be grouped into 3 different areas. So these ones here, which include lung and cervical cancer, it's thought that between 90 and 100% of these cancers could be prevented by having the HPV vaccine for cervical cancer and by stopping smoking. Now, obviously, you can develop lung cancer if you're not a smoker, but essentially you could reduce the risk a lot if people didn't smoke. And then you've got a group of cancers where about 25 to 50% might be able to be prevented. So this includes colorectal or bowel cancer, breast cancer, pancreatic cancer, bladder cancer. So we can modify our risk of colorectal cancer by keeping a healthy weight, not drinking alcohol, and eating less processed meat. And then you've got a group of cancers where we know a lot less about the factors that might cause it or cause them. So here we've got ovarian cancer, where it's estimated that about 20% might be preventable, through to prostate cancer, where we don't know so much about the causes. So here at the moment, the estimate is 0% are preventable. So you can see that it's not uniform across all different cancers. It is quite complicated in trying to understand how we might reduce our risk of each cancer. But a number of organisations do give guidelines for how we can adopt this healthy lifestyle to try and reduce our risk of cancer. So I would, if you are interested in this, I would definitely recommend looking at the World Cancer Research Fund website. They frequently review all the data that comes out every year, and they update their guidelines. So they're a really good source of information to understand really look at what does increase your risk of cancer and what you can do to try and prevent it. And then, of course, Cancer Research UK are a really, really good source of reliable information. And for example, here they suggest 7 ways to cut down cancer. Some of these, hopefully, they'll be obvious after you've seen the risk factors, such as not smoking, keeping a healthy weight, be safe in the sun, drinking less alcohol. In fact, Cancer Research UK have a policy that there is no safe limit of alcohol for trying to prevent cancer, and you'll see why that ties into my talk a bit later with because of its the role of resveratrol and how resveratrol is in red wine. And then, of course, eating a high fiber diet, cutting down on processed meat, and being active, getting more exercise, more physical activity. So these are the ways that they suggest we can try and reduce our risk of cancer as much as possible. But obviously, having this lifestyle is not easy. Otherwise, we would all do it and everyone would be a perfect weight and eat really well. So it's hard to implement these lifestyle and behaviour challenges. And I'm sure we all know someone that has adopted these practices and has had a healthy lifestyle and still goes on to develop cancer. So it's not a guarantee, and it is still very complicated. So on top of these lifestyle and behavior recommendations, we also need additional complementary approaches to try and reduce the cancer burden, particularly for people that might be at a high risk because of genetic factors or other occupational factors, where lifestyle choices and lifestyle changes might not be sufficient to really reduce their risk of cancer. So we definitely need more than this, and we should be doing much better to prevent cancer. So this is where my work comes in, because I work on an area called therapeutic prevention, and that is the use of therapies, so natural or synthetic compounds to reverse, suppress, prevent or delay cancer development to the stage where you've got an invasive cancer. And this isn't a new idea. This is what's done very commonly in cardiovascular disease or heart disease. So this figure here shows how the number of deaths due to heart disease have decreased massively, really, over the last 50 years or so, 40, 50 years. And you can see that they've gone down below cancer, where cancer deaths due to cancer have pretty much stayed quite stable, maybe going down a bit now, but they're quite stable. So this reduction is thought to be due, at least half of the reduction is thought to be due to the use of drugs to prevent serious heart disease. So I'm sure you're all aware of things like statins if you've got high cholesterol, or antihypertensives, so blood pressure tablets if you've got high blood pressure. So there's lots of medicines that they use in heart disease to try and prevent someone that's got risk factors from getting seriously ill. So it's a very common practice. And part of the reason why it's so effective and so commonly used in heart disease is because they have biomarkers, so things that they can measure to show that a person is at risk. And when they take the drugs, when they take the medicine, these biomarkers change so they can show that the drugs are working and they're reducing the thing that is putting them at risk. So for example, blood pressure, if you have a blood pressure tablet, you'll see your blood pressure going down. So you can show that it's working and you're going to be at less of a risk of a serious event further down the line. And the same with cholesterol, you can easily measure it. So these biomarkers are what makes it relatively easy, I think, compared to cancer to develop drugs to prevent heart disease and to also use them. So for cancer, we don't really have these biomarkers. It's much more complicated because cancer is many different diseases, and even for bowel cancer that I work on, there's lots of different subtypes of bowel cancer. It's not just one type of disease. So I think cancer is a lot more complicated, and we don't have these biomarkers, and we really need to develop them. And there is a lot of work going on in this space, but at the moment it's a limitation, and it's what makes it difficult for us. But what this shows is that using therapies to prevent serious disease works. And so we need to catch up with people working on heart disease. So when you develop a therapy to prevent cancer, it needs to have a number of properties. So you want it to work, so it must be efficacious. Importantly, it needs to have no or low toxicity. So this is where it's different or very different from a drug that you're developing to treat cancer. Because I'm sure you will know, if you've known of anyone that's had cancer treatment, radiotherapy or chemotherapy, it's associated with side effects, some pretty nasty side effects. They're not pleasant to take. Even the newer drugs, the immunotherapies, they can still cause significant side effects. But people tolerate that in most cases because they know they need it to get better, to treat the cancer. So the benefits can outweigh the risks and the side effects. But with prevention, because we want to give these therapies to people that don't have cancer, they might be at risk, but they don't actually have it and they might never get it. We can't accept this toxicity. We can't accept serious side effects. So it's really important that the therapies that we develop are safe. They're not going to cause any long-term effects, side effects for the patient, and they're also well tolerated. So they can't even cause like minor side effects like a headache or stomach upsets, because people won't take them every day if they do. So they need to be well tolerated. So this is a key difference between prevention and treatment. We want to know how the therapies work, and this helps us to understand what cancers they might be able to prevent and who we can give them to. This is a quite challenging area, but it's important. And they also, the therapies need to be accepted by humans. So they need to buy into the concept of taking something on a regular basis, probably daily, probably for years. And that's something that people, general public, will need to get their head round so that they are happy to take it. So this is also important. And it's a very different scenario. It's a very different mindset compared, again, to a cancer patient. Ideally, we want the therapies to be an oral formulation, so a tablet or a capsule, to make it easy to take, and they need to be cheap, because if we find things that work, then obviously we're going to want to use them in lots of people, and they will have to take them for a long time, so they do need to be cheap. So because of these reasons, and particularly this requirement for them to be safe, we're limited, or most of the work to date has been limited to 2 different types of compounds. So these are repurposed drugs. So they're old drugs that have been around for a long time and have been used for other reasons. So we know a lot about their safety and what side effects they cause. And then we can repurpose them and use them for cancer prevention. And there's quite a lot of drugs like that. So aspirin is one, metformin that I'll also talk about later. So metformin is a diabetes drug, and that is also being repurposed for cancer prevention. So because they've been around for a long time, been used in hundreds of thousands, if not millions of people, we know a lot about the safety. The other group of compounds that we can use for prevention are dietary related compounds, so constituents of food or drink. And I'm not really talking about obscure compounds from the Brazilian rainforest where we don't know much about them. I'm talking about the ones that are present in our foods that people consume regularly, and they consume reasonable quantities of them. So this would include resveratrol, which are in the grapes and the peanuts here. And we've done a lot of work also on curcumin in Leicester, which is in the spice turmeric. So we know a lot about the safety, or at least we've got no reason to think that they're harmful, and then we can do proper clinical trials to assess safety. So these are the main groups of compounds that we focus on. So novel drugs, like a drug that was developed by a pharmaceutical company, aren't really an option for us for prevention, because when a company develops a new drug, it can take typically 10 years to develop it, and then 20, 30 years to really understand about the side effects that it causes, because we need to get a good grasp on even the rare side effects before we can give it to healthy people to prevent cancer. So novel drugs aren't an option. So and now I also want to now describe some of the challenges and again the differences between developing a drug for cancer treatment versus cancer prevention, just to highlight why this is a difficult area to work in and what we need to be focusing on over the coming 10 years or so to improve the situation. So for cancer treatment, when a pharmaceutical company has got a new drug that they want to test in a clinical trial, they'll take a group of patients, and this group of patients will all have a cancer that they're interested in. And then they'll give them the drug, and then within a relatively short space of time, perhaps months, they'll know whether the drug has worked, because they'll be able to do a scan and see whether the tumor has shrunk, or maybe look at some other endpoints, but they'll get their answer pretty fast to see whether it works. And these types of studies are funded and run largely by the pharmaceutical industry that develop the drugs. With cancer prevention, we need a large group of people, because these are healthy people that haven't got cancer. And then only a small proportion of them are going to develop the cancer that we're interested in. So perhaps, you know, in this group of patients, just the ones in brown are going to be the ones that develop the cancer that we're concerned with. And cancer can take decades to develop. So if we enroll these people in our clinical trial, we might then have to wait for 20 years to see whether they actually develop cancer, so we can see whether our therapies work. So this is how they have done prevention trials in the past, but we now know it's not viable because it takes too long, it's far too expensive, we haven't got the time to wait, and these studies involve too many people. So we need to change how we do them. We need to improve our design of these trials. So there's a number of ways we can do that. If we can identify out of this large group of people who really are the high risk individuals, then we can cut down the size of the trial. And then if we can somehow get some biomarkers or things that we can analyze in the people to show predictive biomarkers to show who is likely to benefit and who is likely to experience side effects, then again, we can cut down this group to reduce it to the ones that are likely to benefit. But this requires that we understand more about the biology of each cancer, and that we understand the mechanisms through which the therapies are working. So this requires a lot of work to get to this stage. Another thing we need are surrogate endpoints. So instead of waiting for cancer to see whether the therapies worked, we can use other endpoints that happen earlier. So one such endpoint is colorectal adenomas or polyps. So these are, they form in the colorectum or in the bowel, and they're little precursors, or they're little polyps. So you can see this growth here in the bowel, and they are potentially precursors to cancer. Now, it's important for me to say here that not every polyp will turn into a cancer, only a proportion of them do. So if you have got polyps, they are removed if they're detected, but it doesn't mean that they're all going to go on to turn into a cancer. But some of them do, and so these can be a good endpoint for seeing whether a therapy works. And this is the endpoint that we use in our trials. And you can get an answer much, much quicker. You can get an answer in a few years as opposed to decades. So we need more surrogate endpoints. At the moment, we don't have many at all in cancer. And the other thing we need are pharmacodynamic biomarkers. So again, markers like cholesterol and high blood pressure that we have in heart disease, that we can measure to see whether the therapies are working. And then if they're working, the person can continue on the treatment. And if they're not working, you can stop and give them something else instead. And you can also use them to optimize the dose and decide what dose is the best to use in the population. So we need to understand all these things much better. And at the moment, there's a shortage of all these different biomarkers to be able to identify who to do our trials in, and to get answers much, much quicker. So this is an area that we're working on. And another key thing which I need to point out is that compared to drug trials for cancer treatment, there isn't much money to be gained in cancer prevention because we need to use old drugs or natural products. There isn't a lot of money to be made from the actual therapies. So consequently, most of these trials have to be funded by charities such as Cancer Research UK or the government through funding bodies that are run by the government, such as the MRC or NIHR. So this is another limitation that makes it much more difficult to do these types of studies. Now, having talked about all the limitations and all the difficulties that we face, I can now go on to a positive, because there are drugs that work. There are only a handful that are currently used, but there are some that work. And one of the best ones probably is aspirin. So aspirin, I'm sure you all know, it's an anti-inflammatory drug. So it's used for inflammatory conditions. It might be used for the treatment of rheumatoid arthritis, or it's used if you've got a temperature perhaps, or as a painkiller if you've got a headache. It's also used to prevent blood clots. So a lot of people might use it, low dose of aspirin to prevent blood clots if they've had heart disease in the past, or if they've had a previous stroke or heart attack. So it's used by a lot of people. And these cardiovascular, or these heart studies where they use it now for prevention of blood clots, the big trials that were done to show this, the results from those trials discovered that not only does aspirin work in the heart disease setting, but they found a relationship between using aspirin and the risk of bowel cancer. So in people that used aspirin in the studies, they had a lower risk of bowel cancer. And on top of that, they also found in the same trials that using aspirin can reduce the risk of dying from bowel cancer by 1/3. So these large epidemiology studies, so epidemiology means you're looking at the large numbers of people, you're looking at population level data, they've been able to show us that aspirin can prevent bowel cancer. And also it looks like it may prevent other types of cancer, but the data for bowel cancer is the strongest. So where it's particularly good is preventing cancers in the proximal colon. So this is the purple part. So this is the first part of the colon. And you can see from these graphs, this is bowel cancer incidence. So in red, it's people that didn't have aspirin, and in blue, it's people that were on aspirin. And you can see the difference between these two lines. So this is the incidence of cancer in the controls and the incidence in those on aspirin, and it's much lower. And the same with mortality. But the key thing is that for these lines to start to separate, it takes about 10 years. So you don't see the benefits of taking aspirin until 10 years after you've taken it. So it takes a long time to see the preventive effects. So not only does aspirin prevent bowel cancer, it also prevents colorectal adenomas or colorectal polyps. And this is important because I'm using colorectal polyps in my clinical trial. And as I've said, they are potentially precursors to cancer. So this is important to show that it can also prevent these precursors. So not only does aspirin prevent bowel cancer in the general population, it also prevents it in a group of patients. I thought my screen had frozen there. It also prevents it in a group of patients that have a hereditary syndrome called Lynch syndrome. So these people they inherit a genetic alteration that means that they have defects in some DNA repair enzymes. So these are proteins in the body that will repair your DNA if it's damaged. And because of these changes to the DNA repair enzymes, they are at increased risk of developing a number of different cancers, including bowel cancer. So you can see from this figure, in the general population, the chances of developing bowel cancer were 5%. So 5 out of 100 people may get it during their lifetime. But with people with Lynch syndrome, the chances are 80%. So they've got a high risk of developing bowel cancer. And a series of trials that have been run out of Newcastle University called the CAP trials. They've shown that taking aspirin for just 2 years can significantly reduce the chances of these people with Lynch syndrome developing bowel cancer. And so this is from data from the clinical trial where they used a high dose of aspirin, 600 milligrams. So these are the people that took aspirin, and these are the people that had placebo or a dummy tablet for comparison. And you can see this is the incidence of bowel cancer. It separates after 5 years. So for the first 5 years, there was no difference. So again, you've got that bit of a time lag before you start to see the preventive effect. But after 5 years, there is this protection, and it lasts for at least 10 years. And there's a subsequent study going on, the CAP3 trial taking place at the moment, where they're looking at different doses of aspirin to see whether lower doses are also effective at preventing bowel cancer in these patients. So I think this is quite remarkable for a group of patients that have such a high incidence of a risk of developing bowel cancer to see such a strong effect of aspirin. And on the back of these trial findings, NICE a few years ago recommend now that people that have got Lynch syndrome should be offered daily aspirin to reduce their risk. So aspirin does work, but it doesn't work in everyone because not everyone was protected against developing bowel cancer. Some patients can't tolerate it. So some people might be sensitive to it. They might be allergic to it, or they might be at risk of bleeding. So one of the main side effects of aspirin is that it causes stomach bleeds or gastrointestinal bleeding. So some people can't take it. And also there's barriers to uptake. So I talked earlier about all the drugs that are used for the prevention of heart disease. And a lot of those drugs are given by GPs. But GPs are much less familiar with the idea of using drugs or therapies to prevent cancer. And so I was involved in a piece of work a few years ago where GPs were given a questionnaire about their attitudes to prescribing aspirin for cancer prevention in people with Lynch syndrome. And it was quite remarkable that a large number of the GPs, so 71% had heard of, or 30% hadn't heard of Lynch syndrome, and a good proportion weren't aware that aspirin could be used to prevent bowel cancer in these patients. And a proportion of them, again, wouldn't have been happy with talking to patients about using aspirin for prevention. And this graph here shows that whether they were willing to prescribe it to their patients depended on what dose was needed. So if they needed 600 milligrams a day, then about 60% were happy to prescribe it. Whereas if it was a low dose, a lot higher percentage were happy. So they still were a bit reluctant to prescribe it for their patients because they're much less familiar with it. So there are these barriers that mean it's difficult for us to use aspirin for cancer prevention. So it's clear that we need safer alternatives to aspirin and other agents that can be used in combination to try and increase the effectiveness of preventive therapies and expand the types of bowel cancers that can be prevented. So we need more therapies that to give us a choice to try and prevent more cancers. And this is where resveratrol comes in as a natural product. So this need for safety that I've mentioned, that drives an interest in using natural products for cancer prevention. And there's lots of different natural products that have been looked at, mainly in the lab. Some clinical trials have been done, but it's not a crazy idea. Some people might think that it's a bit, off the wall, but it's not, because actually, historically, 80% of drugs that are in use originate from nature. So they're either actually an extract, they're extracted from a plant, or they are a derivative of a natural product. So paclitaxel, which is a cancer drug, that is a natural product. And so is digoxin, which is used for heart failure. And then metformin, that is used for diabetes. So this is a derivative. So the natural compound has been modified slightly. And the same with aspirin. So salicylate, which comes from yew trees, that has been modified slightly to generate aspirin. So many of the drugs that we use have nature as their original sources. So now on to resveratrol. So as I've said, resveratrol is present in red grapes and red wine and peanuts and other berries. It's called a phytoalexin, and what that means is that it's formed in plants in response to stress, such as infection or cold weather or other forms of stress. And it's formed in grapes, it's formed under the skin, which is why it's present in high concentrations in red wine, because the skin of the grape stays in contact with the liquid for longer. So actually, in the Western diet, the richest source of resveratrol is red wine, which is good and bad, because obviously for cancer, alcohol is a no-go. So alcohol itself, red wine itself, has no benefits. For all our work has been done with a pure compound. So originally, resveratrol was isolated from a plant called white hellebore back in 1940. And it's also present in Japanese knotweed. So any gardeners out there will be aware of Japanese knotweed. It's present in very high concentrations in that, but I'm not sure if people actually eat it in any shape or form. So in 1992, it came to people's attention when it was proposed to account for the protective effects of red wine on the heart, or the so-called French paradox. And this was the idea that people in France had a relatively low incidence of heart disease, even though they ate a diet that was rich in fat and butter. Now that's since, well, they thought at the time that it was maybe due to the red wine, and maybe it was the resveratrol in the red wine that was being protected, but that has since been shown to not to be the case. They don't have a lower incidence of heart disease, but there is a lot of research looking at resveratrol and its effect in heart disease, and it does play some good protective effects. And then in 1997 came the first demonstration that resveratrol could inhibit cancer development, with a paper in a very prestigious journal called Science. And in this study, they found that resveratrol could inhibit two different types of cancer. So it could develop the growth of precursors to breast cancer in a mouse model, and also in another mouse model, it could protect or could prevent the mice from developing skin cancer. So following on from these first observations, the interest in resveratrol has just increased exponentially. So this is the number of scientific publications on resveratrol since that first paper showing that it could prevent cancer, and it's really increased massively, and it's studied all over the world. So resveratrol has a lot of effects that could be beneficial for the treatment and prevention of many different types of diseases. So amongst its effects are that it has anti-inflammatory actions, it can inhibit tumor development, it acts as an antioxidant, it has calorie restriction mimetic effects. So what this means is that in animal models, and potentially in humans, but in animal models, if you restrict the number of calories that a mouse has, then it will live for longer, and it will have better health if you restrict it slightly. And resveratrol seems to mimic these effects in animal models. It's also been shown, and there was a lot of interest in the media about this a number of years ago, that it can increase lifespan in lower organisms. So this means in worms and flies that are studied in the lab. And it can also, again, in animal models, protect against age-related changes and the prevention of death in animals that were obese. So it can protect against the effects of obesity and a high-fat diet. So on the back of these general effects, there's been lots of clinical trials done on resveratrol, about 150, I think, to date, in many, many different areas. So this just shows all the different areas. So it's looked at a lot in cardiovascular disease and in diabetes, less so in cancer at the moment, and most of that work is from our lab. It's also been studied as its effect, whether it complements exercise or is useful to take it in combination with exercise. And then things like sleep disorders and infections. So chances are, if you've got a disease that you're interested in, resveratrol has probably been tested in it if not in a clinical trial, then definitely in a lab-based model. So it's studied all over the world for lots of different reasons. Sorry, it's not moving at the moment. It's just thinking, PowerPoint's thinking. Okay, well, I can see what the next bit is coming up. So in America, I think resveratrol is used a lot more frequently than it's used here, because you can go down Holland and Barrett and buy it. You can buy it on the internet. It's been shown that of 2/3 of people that regularly take multiple supplements, health food supplements. Okay. Sorry, it's really messed up. I don't think it likes this time on a Wednesday. Right, so yeah, a study in America is showing that that resveratrol is used by 2/3 of people who regularly take multiple dietary supplements. So as I said, it's used a lot more in America than I think it's used in this country, but it is, you know, commonly bought and commonly consumed as a supplement. So in Leicester, we've been interested in developing resveratrol for the prevention of bowel cancer. And that was kind of based on the initial studies that were done in lab models. And so as part of this, we had to do clinical trials in people. And the first stage of developing a therapy for cancer prevention and also for treatment of any kind of disease really is to conduct clinical trials to assess whether the therapies are safe, to understand what side effects they cause and what dose you can go up to, and then to measure how much gets into the bloodstream, how long does it stay in the blood for, and what does it get converted into. Because when we take a drug or any kind of chemical gets into our body tries to get rid of it. So it tries to make it more water soluble so it can be excreted in the urine. So we have to understand what the body does to resveratrol to get rid of it. And because resveratrol is safe, or we thought it was relatively safe, we can conduct a lot of our trials in healthy volunteers rather than in patients. So one of the first ones we did involve healthy volunteers or students in the lab and members of staff in the lab, where we asked them to take resveratrol at a number of doses, and they took it for a month every day, and then they recorded any side effects that they had. So they took it at a dose of 0.5 gram, 1 gram, 2.5 grams or 5 grams. So these are very, very large doses, way higher than anything you could ever get through the diet. And then they recorded their side effects, and you can see at these low doses, there isn't really any side effects reported. But once you get to 2.5 grams, which is a lot of tablets, there was an increase in these sort of stomach and lower abdomen side effects. So a lot of people reported abdominal pain and diarrhea, a bit of nausea at these higher doses. So nothing serious, but obviously, you know, if they're experiencing these side effects, they're not going to continue taking resveratrol. So on the basis of this initial study, we have stated that long-term use of doses of above 1 gram are not recommended for prevention purposes or for any kind of treatment or prevention of disease in healthy people. And we use 1 gram as our highest dose now in our patients and in our work. Now, as I said, the other thing that you do in these early clinical trials is to understand how much gets into the blood and what it gets converted to. And it was known at the time from animal work and cell work that resveratrol, like many other natural products, once it gets into the blood, it very, very quickly gets converted to metabolites. So this is a profile of resveratrol species present in a person that took 2.5 grams, and this is 1 hour after they took it. And what this shows is that there's only a small amount of resveratrol left. Most of it has been converted into these peaks here, which are resveratrol metabolites, which are more water soluble, so the body can get rid of them. But as far as we knew, these metabolites don't have any activity. What we want is to get high levels of resveratrol into the blood. So it's clear that it gets it gets metabolized very quickly and the levels of resveratrol are low in the blood. Now, we've also did studies in patient population. So because we're interested in bowel cancer, we wanted to know how much resveratrol gets into the bowel tissue to see whether we can get enough of it in there for it to have an effect. So we did what's called a window trial, where we recruit patients that are having surgery for colorectal cancer, and we take advantage of the window between them being diagnosed with cancer by having a biopsy and having surgery to have the cancer taken away. In that window, we can give them resveratrol, and then when they have their surgery, we can get a piece of the normal tissue or the cancer tissue if it's spare, not needed for pathology, and we can analyze how much resveratrol is in there. And that's what we did here in a study in patients at Leicester Royal Infirmary. And this is a pattern of resveratrol species in colorectal or bowel tissue. And actually we found in the bowel tissue, there's pretty high levels of resveratrol. So unlike the plasma, the levels are much higher. In fact, they're 70 times higher than we saw in the plasma. So we thought this was really, really good news because we want to target the bowel. So that was a big positive. And at the time, a lot of people that worked on resveratrol were kind of worried or confused as to how resveratrol might be able to have an effect, because it does get metabolized to these other species so quickly. And so there was, there were questions about whether these metabolites might have activity themselves that could be beneficial, or whether they might be able to regenerate resveratrol in the body. And I'm not going to show you the data, or show you our results, but we found that this is resveratrol, and then it gets metabolized to a number of species, but one of the main ones is resveratrol sulfate, and this yellow circle here is showing you the sulfate group that gets put on by the body. And we found that this compound, as well as being excreted, it can also get into cells, inside cancer cells, and when it gets inside the cells, this group comes off and it regenerates resveratrol, which is then able to have anti-cancer effects in the cancer cells. So we showed actually for the first time that these metabolites, yes, they're formed in high levels, but they could still be useful. They can still contribute to the activity of resveratrol. Now we've also, I was interested in trying to understand what dose of resveratrol we should use in our clinical trials, because obviously we became interested in it as a natural product. And a lot of people that work in this area on natural products, the compounds first come to their attention because of epidemiology data. So this population level data showing that populations of people that consume diets containing these compounds have lower incidence of certain diseases, such as a specific cancer. And that suggests that low doses or low amounts of these compounds consumed over long amounts of time are what are having the protective effects. But then researchers go on and do clinical trials using much, much higher doses, kind of assuming that more is better. And we did it ourselves. I showed you the doses that we used in our previous clinical trials. But at this stage, I was interested in asking whether a dietary relevant dose, a kind of dose that you could get through a couple of glasses of red wine, might have effects. And are the effects better than the effects that you can get at a 1 gram high dose? So the first thing we had to do was see whether this low dietary dose, which is 5 milligrams, can actually get to bowel tissue. So we did a study in patients, again at Leicester Royal Infirmary, where we gave them a low dose of 5 milligrams for 8 days, or 1 gram, which is 200 times higher, prior to these patients having colorectal or bowel surgery for cancer. And the last dose we gave them was carbon-14 labeled. And the reason we did that was so that we could use a super sensitive technique for measuring the resveratrol in the tissue, because we weren't sure we were going to detect anything with this low dose. And the technique we used was called accelerated mass spectrometry, which is the method used for radiocarbon dating. So you might have heard about it for radiocarbon dating of like religious artifacts such as the Shroud of Turin or the Dead Sea Scrolls. Or dating old trees, things like that. So it's basically the most sensitive method for detecting trace amounts of the radioisotope C14. But because it's so sensitive, we only have to give a tiny amount of radioactivity to our patients. So it was very safe. The amount we had to give them in our study was equivalent to the amount in about 200 bananas. So it is a safe method. So we gave them these 2 different doses, and then at the time of surgery, we collected tissue and blood and analysed it by AMS. And what we found was that we could detect it after both doses in the tissue. So the low dose was able to generate detectable levels in the tissue, which we were very happy about. And the concentration that we detected was dependent on the dose. So unsurprisingly, there was about a 200-fold difference in concentration, and also the time. So by this, I mean the time dependent, the concentration was dependent on what the interval was between taking that last dose and having surgery. So the longer the time interval, the lower the concentration, which makes sense. So this just shows that we were able to detect a peak of resveratrol, so resveratrol rather than metabolites, in the mucosa. So this is the inner layer of the bowel tissue, and then in the muscle layer which surrounds the bowel, in both these layers in a patient that had the low dose. So this was really good news. And then we could use this information, the concentrations that we detected, we could use that information in our lab studies and make sure that we treated our cells in the lab with these kind of concentrations to make our lab work relevant to patients. Oh, it's stalling again. I'll just wait. So having shown that resveratrol can get to the target tissue that we want it to, the next question was whether it might be able to actually have benefits and prevent, sort of inhibit the cancer development process in a mouse model. So to test this, we used a model called the APC min mouse, which is a model of human colorectal cancer, bowel cancer. And the mice develop lots of little polyps in their intestine. And so you can give them a normal diet, or you can give them a diet with resveratrol and measure the number of polyps at the end of your study to see whether the resveratrol is able to inhibit polyp development. Good, it's working. And so this is some results from some experiments that we did where we gave resveratrol at the same 2 doses as we did in our clinical trial, and also a control group to see whether it could inhibit polyp formation in these mice. So this is animals on a standard diet. So these are control mice. This is low dose and high dose resveratrol. And so each circle or square is a mouse, and it's plotted against the polyp number per mouse. So here, basically, the high dose of resveratrol had a small effect. So it was able to reduce the number a bit, but not by much. And then this is polyp volume. So this is the total volume of all the polyps added up in each mouse, and there was no effect there. So these are the 2 standard ways that we would test to see whether a compound was having any benefits. So that was on a normal diet. But at this time, there was a lot of evidence coming out that resveratrol could protect against the effects of a high-fat diet. So we did the same study in our mice using a high-fat diet. And amazingly, both doses worked, but the low dose, this tiny dose, was able to inhibit polyp development in the mice more effectively than the high dose. So this was both polyp number and polyp volume, which we weren't expecting. So there's an unusual dose-response relationship going on here. And then we did some experiments to try and understand what was going on. And it seems like the high fat is causing more polyps to develop in the mice. So it causes the cells to divide more and to proliferate more, and this leads to more polyps. And we were able to show that resveratrol could cancel out these effects of the high fat diet. And then we did a lot of mechanistic work to try and understand how resveratrol was working using our lab model. So we used cells that came from the mice, came from the intestinal tissue of the mice. And we did a lot of experiments to understand what mechanisms, what processes were affected by resveratrol. And we found that it affects a protein called AMPK. And AMPK is important because it regulates a lot of how energy is used in tissues or in cells of the body. And so we found that resveratrol could activate AMPK, which means that it was able to shut down processes that generated a lot of the energy and a lot of the requirements that cancer cells need to proliferate. So it could block protein metabolism, and it could block the production of fats that the cells needed to divide. So this was a potentially beneficial effect. And we found, the important thing we found here was that, like we saw in the mice, we had this unusual dose response where the low dose was more effective than the high dose. Sorry, it's stalling again. And the upshot of these effects was that resveratrol was able to cause two processes called senescence and autophagy, which basically means kind of putting the cells to sleep. So they're still alive, but they're not multiplying, they're not dividing. Or here, autophagy is where they kind of self-eat their contents because they think there's something wrong with them. So these two processes, resveratrol was able to cause these processes to happen. And whilst the cells are doing this, they're not able to divide. So again, we take these as good things that are useful for cancer prevention. And there were stronger effects at the low dose. We then wanted to know whether these effects might translate to human tissue. So we did some experiments using tissue that we got from patients that were having surgery, and then we take it back to our lab and we can treat it with resveratrol in a dish in the lab to try and understand how it's working. And what this shows is that resveratrol activated the same pathway. So it was able to activate AMPK in the human tissue, and it could cause autophagy and senescence in the human tissue. And again, the low dose had stronger effects than the high dose. And then this is some data from our clinical trial where we gave our patients the C14 labeled resveratrol, and we analyzed the tissue for the presence of antioxidant markers. So these are this is NQO1, which is a protein that is increased in response to oxidative stress. So it tries to protect cells if there's a lot of oxidative or reactive oxygen species around. So if this increases, this is protective. And we found that the low dose increased the levels of this protein, but the high dose didn't. And again, this is an indicator of a reactive oxygen species that was increased at the low dose, but not the high dose. So all of this information together is telling us that the low dose of resveratrol is having effects in humans that are potentially beneficial for preventing cancer. Oh, I'm sorry, my slides keep sticking. So you might think, based on all of this, we should be pursuing the low dose, the 5 milligram dose of resveratrol, and forgetting about the 1 gram dose. But we are still pursuing the 1 gram dose because I think, first of all, the two doses have different mechanisms of action. So I think the low dose affects the bowel tissue, whereas the high dose affects what's going on in the blood, and this in turn can affect the tissue. And I also think that you might need different doses of resveratrol to prevent different subtypes of bowel cancer. So I mentioned earlier that bowel cancer isn't just one type of cancer, it comes in different types. And for one type that is less common, that is driven by an oncogene called BRAF, I think the high dose is needed to prevent this, because we've got some information from a different animal model, different mouse model, where the high dose is what works for preventing cancer in that model, and the low dose doesn't. Oh, sorry. It's whirling round, so it hasn't completely crashed. Oh no, it wants me to restart the program. Alison, any suggestions? Shall I close it and start again?
It's entirely up to you. I wasn't sure how much longer you have with your presentation, or whether, because we have come to 6:30, so I don't know.
Or is it maybe another 10 minutes? I've got to get to the good bit now.
Happy to. That's not
okay.
a problem at all. We'll keep our fingers crossed for you.
Okay, I'll tell you what, I will restart it.
These things happen at the best of times, don't they? If we can just thank everybody in the audience for bearing with us, that's really helpful. Thank you.
Don't often use Zoom, but
if anybody does have a question that you want to pop in the chat whilst Karen's sorting her end out, by all means do so, and we'll see what we can pick up from there.
Right, can you see it?
Yes, we can.
Right. Okay, so yeah, so we're at the moment we're still interested in both doses. So what we wanted to do next ultimately was see whether resveratrol works. Can it prevent colorectal polyps and cancer in humans? But to do this, there wasn't an easy way. There wasn't a simple way of testing it in patients. So we had to develop a brand new clinical model for testing preventive therapies within existing NHS practices. And this required a new collaborative approach. So I developed a new UK group involving lots of different people from different backgrounds that could work together to develop a trial called ColoPrevent. So to test resveratrol, we have to have a group of patients that are at high risk. And so we are using people that are identified as high risk for developing polyps through the bowel cancer screening program. So some of you may know that when you get to 60 years of age, you get given through the post a fecal immunochemical test for testing for the presence of blood in feces. So this is for bowel cancer screening. And if you get a positive test, you may be invited for a colonoscopy where they'll search for cancer. And if you have got cancer, then obviously you'll be treated. But there's also a group of patients that may have polyps. And if you've got polyps, they'll be taken away at this point, they'll be removed. But then patients may be classed as low risk for the polyps coming back or for cancer, in which case they're fine. They just carry on their way and pick up with general screening again. Or they may, oh, can you see the, or they may be classed as high risk. And if they're high risk, then they'll be asked to come back at 3 years for a surveillance colonoscopy. And at this point, about 60% of patients might reoccur with another polyp. So this group of patients, they're really good candidates for prevention to try and stop these polyps from coming back and from cancers from developing, and also as a model for us to test our preventive therapies in. So this is our patient population that we're using for ColoPrevent. And so in developing ColoPrevent, we decided not just to develop a trial for resveratrol, but if we were going to this effort, then we wanted to develop a larger platform that enabled us to test other therapies as well. So it's a new way of testing preventive therapies. And we also wanted it to be sustainable long-term infrastructure that enabled us to continually test in the future whenever any other good therapies became available. So, or even things like an exercise intervention or a low-calorie diet that could also be tested within this clinical trial structure. So in the first part of our study, first part of the ColoPrevent platform trial, we're actually doing 2 studies. We have a phase 3 clinical trial and a phase 2 clinical trial. So in the phase 3, which is a more advanced type of testing, we're actually testing aspirin versus aspirin plus metformin. So this is where aspirin comes into my work, because we think these patients should actually be on aspirin anyway. And then we're asking, if you bring in metformin, does that give more benefits? So the reason for including metformin is because there's a lot of epidemiology data to show that metformin can prevent bowel cancer, and also a study done in Japan to show that it can prevent polyps in a similar population in Japan. And then, of course, we're looking at resveratrol, where we're going to be comparing that low dose, 5 milligram, versus a 1 gram dose versus placebo or a dummy capsule. So this is an outline of the trial. So this is the main phase 3 study. So people can go into that study if they're not on aspirin or metformin already. And then if they satisfy all the inclusion criteria, they will be randomized to take low dose aspirin or low dose aspirin plus metformin. And they'll take this for 3 years, and then they'll come back for their standard surveillance colonoscopy at 3 years. And our outcome, so what we're looking to see whether it's changed, is polyp number per patient, per participant. And then we'll have other outcomes around the size of the polyps and where the polyps might be in the bowel. And then we've got the signal-seeking sub-trial where we're looking at resveratrol. So any patients that are already on aspirin or metformin and can't take part in the main trial, or if they don't want to take aspirin or metformin, or they can't take it because of other conditions that they've got, then we can offer them to take part in the resveratrol study. And because this is a signal-seeking study, it's a smaller study and it's a shorter study. So in this study, patients will be randomized to placebo, the dummy tablet, resveratrol low dose, resveratrol high dose, and they'll take this for a year, and then we'll have the same outcome. So we're looking at polyp number per participant and the size of polyps and where they are in the bowel. And then after, when we do this surveillance colonoscopy at 12 months in these patients, they'll then continue in the bowel cancer screening program as normal, and they'll have their normal colonoscopy at 3 years. So there's a number of innovative features around the ColoPrevent trial. What we're trying to do is take on board all the advances in trial design that take place in cancer treatment setting and move this into prevention. So it's the world first prevention trial platform. What we've tried our very best to integrate it into standard NHS practices to make it easy for the clinicians and easy for patients. And also, as I've said, it can respond to any new developments, and we hope to add in things like vaccines and dietary interventions in the future and keep it running. So there's always going to be something going on within the bowel cancer screening program setting for testing preventive therapies. And we also have a lot of embedded laboratory work to try and help us understand who should take what therapy, so that we can ultimately tailor the therapy to the individual. And this just shows some of the biomarker work or the laboratory work we're doing in Leicester. And I just really wanted to highlight a couple of things. First of all, I'm really interested in whether metabolic status, so the metabolic health of the person, influences whether the therapy works, because there's some evidence that resveratrol, aspirin and metformin might work better in people that have got metabolic disturbances. So maybe people that have got pre-diabetes or diabetes, or maybe overweight, it might work better. And then the other interesting thing we're going to be looking at is the gut microbiome, to see whether the profile of bacteria or the microbes that people have got in their gut predict which therapies are going to work. And then if changing the microbiome by the therapies is part of how they do actually work, it could be part of their mechanism of action. So we'll be looking at that over the course of the study. So the study has opened. We're going to open it across 60 sites. It's a 10-year study funded by Cancer Research UK. And we're going to be recruiting a total of about 1,400 patients. As I said, the study is open. We opened at Kingsmill Hospital, which isn't too far away, in Mansfield. And so we're hoping to actually recruit the first patient very soon. And when that happens, I've got a bottle of wine in my cupboard that I've had for almost a year, and I'm waiting to open it for when the first patient enters the study. So I'm hoping that that's going to be very soon. So I will leave it there. I just want to say that research, particularly clinical trials, is teamwork. So as a result of setting up ColoPrevent, I've had to work with a brand new set of people that I've got to know. It's taken about 5 years to get to this stage. So there's a lot of key collaborators involved in this. And then the clinical trial group at Leicester University who are running the trial. And then a whole team of lab people have contributed to all the lab work that we did to get us to the stage where we could design and run ColoPrevent. So this is just, I can't go through everyone, there's too many, but this is a picture of the lab from a few years ago and some of our key collaborators. And then our funders, the Cancer Research UK, have largely funded all our work on resveratrol. We're supported by the Experimental Cancer Medicine Centre at the University of Leicester and Hope Against Cancer, which is a local charity. So yeah, I'll leave it there. Sorry for going over time.
Thank you very much, Karen. That's fantastic. Really enjoyed the talk. Thank you.
Yes.
So if any